Hello, I'm Matthew Lettow. I worked with Kyle Palos in the Nelson lab to study mRNA modifications across Arabidopsis and sorghum. So, like DNA, RNA can be chemically modified. The most common mRNA modification is M6A, and in specific studies, it's been shown that specific sites can greatly change the folding of the resulting mRNA or change the translatability. But there's still a lot that needs to be learned about it. This research focused on the question of, do different plants do their M6A in the same way? So we chose two very different plants, Arabidopsis, a dicot, and sorghum, a monocot, in order to test this out. And we sequenced them with the newest nanopore sequencing chemistry and predicted it with their Dorado. And it turns out that, yes, these two very different plants that we didn't expect to do their M6A in the same way do have very similar M6A epitranscriptomes. So you can see here on this Venn diagram, this is all the genes that were expressed in all three samples, and 45% of them were methylated in all three samples. We also looked at where on the transcript these methylations are occurring, and you can see that they're occurring in the same spots. In all three of our samples, they're occurring in the 3' UTR and right before the stop codon. And then I lined them up specifically to see where they were in relation to each other, and I found 3,647 sites in Arabidopsis that had a site in sorghum at the same spot or really close next to it. This here is just one of those sites that I lined up in a genome browser, and you can see that the purple M6A sites line up perfectly. These sites will be great potential candidates for future research on what these M6A are doing, because these two very different plants have them in the same spot, so they must be providing some beneficial effect for these two plants to be using it in the same way. Thank you very much. That's my research.
Thank you. Very nice. What was your favorite part of this project?
Favorite part of this project? I learned a lot about R, and I really enjoy making graphs in it now. Before this REU, I dreaded making graphs in R. I was like, please let me use Excel for this because I understand that. But now I get R, and it's so much better.
Thank you so much.
Thank you.
Nice work.